In this Happy Scientist seminar, Zhu and I will introduce you the Annotation Query, which is an integrated and interactive platform for large-scale genetic variant annotation. I'll first tell you our motivation behind building this tool. Then I'll give you an overview focusing on the difference between this tool and other tools out there. Zhu will tell you more about the implementation and give you a live demo. So in this post-genome sequencing era, scientists who study diseases are more interested in finding out whether there is any association between underlying genetic traits and the diseases. This schematic diagram shows an association study in cancer research, which tries to see whether there's any variance with higher frequency in cancer patients than that in the control group. If you are lucky enough, you'll get this beautiful Manhattan plot. But the question is this the end of the game? Of course, the answer is not. Getting a list of variants is the beginning to answer the question. What you want to know is whether these variants are related to any functions, such as pathways and processes in the cell and tissue. Ultimately, we want to understand the function they are associated with that will help us to understand the mechanism of the disease. In order to answer these questions, we have to first link the function to the variants, a process we call annotation. For decades, functions have been studied in genes through wet lab experiments such as biochemistry, molecular biology, and cell biology. There are dedicated resources with experts who rigorously go through the literature to assign functions to the gene, a process we call biological curation. These resources include Gene Ontology Consortium and various pathway databases. So if somehow we can map the variants to the gene, we are able to associate these functions to them. How do we do that? We first map the variants to the DNA location using the genomic coordinates. We can then determine whether it is in the gene coding region or non-coding region. If it's in the gene coding region, then it's simple. We can map to the gene directly. However, only 1 to 2% of our genome falls into this region. More than 98% of it is in the non-coding region. In this case, we can still map these variants to the gene if it is if they fall into regulatory regions of the gene, such as promoter or flanking region of the gene, or methylation region, or enhancers. But one thing I want to point out is a majority of the variants still cannot be mapped to the gene in any case. Variants are also associated to, can be annotated to allele frequency. Some variants have been studied in and shown to be associated with specific diseases, and they can be mapped or annotated directly to these diseases. In the past decade or so, many variant annotation tools have been developed, but most of them are focused on data highlighted in blue in the middle here. Most of them require some bioinformatics skill in order to set them up and use them. There's no comprehensive functional annotation data included in those annotation tools. You will have to use separate tools to obtain these functional annotation. Although enhancers can be annotated to the variants, there's very little functional data linked to these enhancers. So if you want to run any annotation using these tools, you have to set each individual tool by yourself, and that's a tedious job. And also, it takes a lot of upfront overhead in order to do that. So this is just a summary of what the current situation is. for these tools. Some of those I already mentioned in the previous slide. Most of the current tools require computational and bioinformatics skills. It is usually difficult to set up and use them in a standardized way. You usually need to do it individually. Each tool may have different system requirements, support different genome build, and even different ID types. And to keep them up to date could be a nightmare also. It is possible that these tools are used by a core facility or for research groups that use them on a regular basis. It is not cost effective if you just want to annotate a few variants. And most of these tools don't have the functional annotation capability built within the tools. Even if they have functional annotations, the annotation tend to be very sparse and often outdated. And there's no functional annotation to enhancers. The motivation for us is to build a comprehensive and easy to use variant annotation system with the most up-to-date functional data that is accessible to researchers with different research background. Here comes the annotation query. This is the homepage of the system. You can access the system through this URL, www.annoq.org. Zhu will tell you more about how to use each individual tools within that platform. So here are some highlights of the platform. First, it is easy to use. No set of requirement and no computational skill is needed. And there are multiple ways to access the data. And one is the web interface. The second is a programming package, such as an R package that you can embed in your script. And the third is, if you are familiar with Unix or Linux, you can access through command line. And this will allow you to access the data through this sophisticated API. Comprehensive functional annotation are included in the package. These include gene ontology and pathway annotations. These data are regularly updated. Last, the platform includes functional annotation to enhancers. This is done using the PANTHER and Database, which stands for Predicted by Experimental Results, Enhancer Gene Regulation, Enhancer Gene Relationships illustrated by a Nexus of Evidence. This is a platform, this is a database that is developed by Caitlin Mills, a student in the group. She'll tell you more about this project in an upcoming Biostats seminar. This is what AnnoQ works. It leverages the existing tools developed by the community and adds on additional functionalities to make it easy for the user to use it. First, large variant data sets are pre-annotated and stored in the database. And we took care of all the headaches to set up the tools, resolve discrepancies, and keep all the data up to date. The annotation now becomes a simple query. It is fast and easy. The second is the functional data are integrated into the part of the pipeline. And the third is the enhancer annotations can be easily accessed through this pipeline also. Overall, this becomes a one-stop shop. And these are the annotation resources included in the AnnoQ platform. Most of them are available through the existing tools out there. The ones in red have been integrated to the workflow by AnnoQ. I'm not going to go into detail of each one of them, but if you're interested, you can obtain more information using this URL link in the bottom here. The backend of AnnoQ is a large annotation database, which contains pre-annotated variant data. Currently, about 40 million variants from haploid haplotype from Haplotype Reference Consortium are included. They are annotated with sequence feature, diseases, allele frequency, etc., using WGSA shown here. And they're shown in blue, all these annotation data. And they are also annotated with functions through PANTHER and Peregrine. At the end, the annotation database is a huge matrix with 40 million rows of variants and over 1,000 columns of annotations. And this diagram shows the overall architecture of the platform. The annotation database is built with Elasticsearch framework with the data I mentioned in the previous slide, and can be accessed by users through this API. There are 3 different ways to access the data. The first is through a web interface. You can annotate up to 10,000 variants using this interface, and you can view the results and you can download it, of course. You can also query, for example, by genome coordinates and gene identifiers, and retrieve a list of the variants associated to these query terms using this interface. More importantly, you can use this interface to determine what type of annotation you want to retrieve. Like I said before, there are more than 1,000 annotation types in the database. Not all of them are pertinent to your analysis. You can select the ones that tailor to your analysis and store the selection in the configuration file. The file then can be used by the other 2 methods to annotate the data. Now, the second way to annotate data is through the command line. And this is a very simple way, and you can annotate more, if you are annotating large number of variants, more than 10,000, you can use this method. The configuration file can be used to retrieve the desired annotation data here. And the third way to annotate the data is through a programming package. We have developed the R package so that it can be embedded in your R script and retrieve annotations on the fly while you analyze the data in R. Ju will give you a live demo about how to use each one of them. I would like to thank everyone who helped us to build this tool. Particularly, I would like to thank Tremaine for helping us on the UI development, and Caitlin for building the Peregrine database. And also, this work is funded by a PO1 grant from NIH. Now I'm going to pass it to Ju, who will tell you more about the implementation and give you a live demo.
I will introduce a little bit about how we implement AnnoQ, and then use several live demos to show how can we use AnnoQ. AnnoQ is an integrated and interactive platform for large-scale genetic variants annotation. Basically, we pre-annotate those genetic variants and store them in a search engine so that people can retrieve the annotation information with multiple ways and perform filters on the variants dataset. This is a main website for AnnoQ. The address for this website is annoq.org. Now I want to talk a little bit about how we implement AnnoQ, because this is very important for we to understand the infrastructure of AnnoQ and get some idea about how can we use AnnoQ. Here is a graph I used to show AnnoQ infrastructure. Our AnnoQ has 3 main components. The first one is AnnoQ website. The second one is an R package, which is called AnnoQ R. It acts as a client for AnnoQ API. And the final part is our AnnoQ API, which is a real data provider. AnnoQ website and R package communicate with AnnoQ API through HTTP JSON request. Start with the AnnoQ API. The backend for AnnoQ API is Elasticsearch. It is a search engine which provides multiple query functions for AnnoQ. We store our pre-annotated variants in this Elasticsearch. I skipped the part which we processing the data and perform the annotation steps here because this part won't influence the usage for AnnoQ. You can imagine Elasticsearch as a large table which has thousands of columns. Each column is an annotation item for the variants. People can perform query based on some certain column and only retrieve data with the columns they interested. Besides the Elasticsearch, we have an additional wrap layer. This wrap layer provides functions such as user control and gene name mappings. It makes the data structure in Elasticsearch more simple. With this Elasticsearch and the wrap layer, we have the flexible to edit some additional annotation items in the future, and we can expand our dataset size to billion level easily because Elasticsearch is well horizontally scalable. And when we go back to AnnoQ website, it has several pages, which is documents for our AnnoQ. We organized all of our documents and descriptions in this website so that people can have an endpoint to reach all of the resources provided by AnnoQ. Besides those documents, it also has an interactive query part. It is a graphic interface for users to learn the data structure. Besides this, it can also generate configuration files. These configuration files can be used by R package or other programming language which use our AnnoQ API. And then we go back to our AnnoQ website. When we go to our AnnoQ website, first we will see this welcome page. In the navigation bar, we have the interactive query, we have our data access, which is a document for our AnnoQ API, and also for our annotation document, which explain each annotation items here, and the about part, which is a fully description of our AnnoQ. We also list the introduction for all of the three AnnoQ main components here and added the links for our three components here. Following is a step-by-step graphic interface tutorial. By reading this tutorial, users should know how to perform queries with interactive query part. It describes different types of query we can use here and also describe how we can export our configuration file and also something about the result. Let's go to the interactive query part. In the interactive query part, first we need to choose the input type. Here we have 5 input types. The most simple one is chromosome. This is you input a chromosome coordinate and retrieve all of the variants inside that chromosome coordinate. Here I choose chromosome 18 and from start point 1 to the end point 500,000. And then I need to select annotations. This is a list of all the annotations we have, organized in a tree structure. For example, I'm only interested in ANNOVA annotations, so I can select basic information and ANNOVAs here. And then I can click on submit. The AnnoQ graphic interface will automatically retrieve the variants inside this genome chromosome, genome coordinate, as well as all of the annotation information belongs to the ANOVA here. And you can see the result as a table at the right side. You can scroll to the right side to see each column. Some columns may be empty, and some columns, for example, this ANOVA ensemble transcript ID may have multiple transcript IDs, and each transcript ID is separated by a tab here. We will double click on one row. All the information in this row will showed in a summarized panel here. And this will skip all the empty column, so you can have a more concise summarize for this variant. You can also download all of the variant in the current query with download button here. You click on the download button, and when the download data is ready, you will see a dialog panels here. And from the link, you can download the all of the variants inside this query. This is quite large, and it's definitely larger than the variants can show in the graphic interface. And the second one is VCF file. This is kind of like multiple queries. You provide a VCF file. Here we have a sample VCF file. For VCF format, you can refer to the formal definition on website. And when you have a VCF file, you have multiple variants inside this file. When you click submit, the VCF file is uploaded to the server, and then you can see all the variant with annotations inside this VCF file. The same thing when you double click on one row, you will see a summarized panel at the right side. And the next one is gene ID query. So when you choose gene ID, you can provide one gene ID here. And when you click submit, the AnnoQ will automatically mapping the gene ID with PANTHER API to a standard PANTHER gene ID. Here we use HGNC ID and UniProt ID. And when mapping to existing HGNC ID and UniProt ID, the gene ID is converted to the genome coordinate, and then we will retrieve all the information from this gene, from this genome coordinate. And the next one is RSID. RSID, you can directly use RSID to retrieve only one variant, and the same thing for the tables here and the summarized panel here. And the last one is a general keyword search. So you can provide any kinds of keyword you may think is you are interested in. For example, I use signaling by GPCR here. And AnnoQ will automatically trying to match this keyword ID, this keyword to multiple annotation columns and retrieve all the variants which is related to this keyword. And because it use many types of annotations here, this one is a little bit slower than the previous one. And the same thing, you can download all the data from this query by using the download function. And next one, we are going to see the AnnoQ API. First, we go to the AnnoQ welcome page, and you click on the data access. You will redirect to the AnnoQ API document. This document I show how the structure for the request, how the structure is a response. Because our AnnoQ is based on Elasticsearch, it has all the functions Elasticsearch may have. And for those functions, you may refer to the Elasticsearch official website. And in this document, we mainly state things for our RAP API additional functions. Besides the documentation, I also provide several bash scripts as examples to show how you can use this AnnoQ API. and we can download all the script in our computer and run those bash API to see what is the response. And for looking at those bash script, we will know what is the request format. I have already downloaded them here, and these are the list of all the bash files we have. One thing special is a host script here. This is for storing the host name in a variable. For example, if in the future we changed our host name, and you only need to modify the host here, and all the other script can still run. Most basic one is a region, which is quite simple. Just perform query inside the region. Looking at this bash script, I use curl here to send a post request to our host name with some certain endpoint. This should be a HTTP JSON request with a JSON body, has this certain structure. In the JSON structure, we indicate that we want chromosome 2, and the position should greater or equal than 10, little or equal than 20,000. Let's run this script. So you will see the response. And if we use some software to print those response in a more readable way, you will see it has some things state the status and the time used for this query, and then following all the data was in this heights field, and each one should has all of the annotations in this data set, because we didn't specify which column we want. And if we want to specify which column we want, you can use this region underscore field. So it has some additional request parameters. So here is underscore source, and this means I only want the response with this ANOVA ensemble gene ontology biological process list. So let's run this script. So you can see the response is much shorter than before. It is because we only retrieve the one column here, and some variants may not have this column, so that column doesn't appear in our response. And next one is the size. So we can indicate, for example, if response has multiple pages, we can indicate the number from and the size. It will control which part of the response we want. And a more important one is this download script. This shows how you can use the curl post request to retrieve all the result from one query. This is the same thing as a download button in the graphic interface. And also, for example, we have the RSID query part. This is simpler than the previous one. They send a GET request with RSID. And by modifying this script, you can retrieve multiple RSID with one script, which is not provided by the graphic interface. And then we go to explore our R package. So you can reach the R package from AnnoQ welcome web page and click R package here. It will redirect to you to the R package GitHub page. And you can install this R package from GitHub by using DevTools. And I also provide several functions to show how you can use R package to retrieve data from AnnoQ API. I copy those examples here. So first you need to load the AnnoQ library. And we have provided several functions act as graphic interface functions, the same as graphic interface functions. For example, you can reach all the variant from the certain region query, and you see all the result is stored in a list format in this variance variable. And if you have more customized requirement, you can use this R client in another way. For example, all of those functions named with underscore is kind of like inner functions, and functions named as this camel style is complete functions. For inner functions, it doesn't complete the function, but it is a component. You can combine them together to reach some certain functions. If you want to query variance with only one column here, you can use those component functions. First, you need to init a query body, and then you added this exist filter to the query body, and then you use a perform search function to send the query body to the AnnoQ API. You can get the variance information in this variable. And the same thing for other kind of filters you can use. This is an example shows how you can use term filter. Term filter means that you can specify some term and only retrieve rows with the columns equals to the value you provide here. For example, I init empty query body here. I init two term filters. The first one, I want to retrieve SNP-F ensemble effect as intergenic region, and the second filter is chromosome. I only want variants from chromosome 20, and I added those two filters to our empty query body and perform this query, and you get the result. And another part is that you can use the configuration file you download from the graphic interface. So I have already downloaded a graphic interface configuration file here. We can take a look at this configuration file. So it means that I want the annotations from ANOVA ensemble and all the other information belongs to ANOVA part. And we can add this source parameter to our empty query body and perform search, and you will get all the ANOVA annotations with variants which can retrieve with this query. And the RSID is a ready function, so the function here is named as a camel style, and you can just provide one RSID and got that all the information inside that variant.